All right, everyone, welcome back. So we have finished 3 modules. We're on to now module number 4, and we're going to look at the Simon task and the Simon effect. So the task refers to the procedure that the participant does, and then the effect is, what occurs in the data. So it's kind of same thing planned for the next 2 modules. It's just new content. This kind of still falls within the realm of compatibility. Actually, it's kind of the very last part of that reading, or on its own page, kind of depending on which PDF you read. But this is going to have some similarities to compatibility, and we'll actually see the dual process model again. Any questions about kind of where we're headed with 2 more modules? Yes.
We have this one and one more.
Yep. So this is 4, and then 5 is going to be, I guess, more of a true experiment, in that we're going to do something that, so 1, 2, 3, and 4, we've been trying to replicate famous experiments. Number 5, we're going to run our own little experiment where I don't know what's going to happen, and I'm curious to see what will happen. Probably won't have the profound impact of something like the Simon task, but it will be our own little experiment. So I think there's 11 of us, 10 of y'all. Is that right? 5, 6, 7, 2, 4, 6, 8, 9, 10. So let's see. I knew one person was going to be away. Madeline's not here, right? Okay, I knew about Madeline. I was recording for Madeline.
Yeah, Kelsey.
Kelsey. Okay. Because we're not. Are we down to 12? Yeah, we were 13. We're down to 12. Okay. But no Kelsey, so hopefully she's okay. Has anyone seen her today? No. Okay. All right, let's talk about the Simon task. And there wasn't a lot in the textbook about this, so I'll give you some background. And actually, I'll give you some historical background. So it's named the Simon task after J. Richard Simon, who discovered this. Well, he designed the task and discovered the effect, the Simon effect. So it's named after him in his honor. I'm not quite sure about the second author on this first paper. I don't know if it was like one of his students. Or I'm actually not sure. I'll have to look up what the story is about the second author. So I have a quote here from you, actually from Professor Simon himself, about designing and discovering the Simon effect. So he said, "My research on the so-called Simon effect began by accident. I was originally interested in the phenomenon of hemispheric dominance for speech." So with speech, trying to get this right, your left hemisphere is dominant for speech. So if we disrupt your left hemisphere, you can actually lose the ability to speak. But you can still sing, because singing is controlled in your right hemisphere. That's for most right-hand dominant people. Not all, but most. For left-hand dominant people, some lefties, their hemispheres are the exact opposite of righties, and others are the same as righties, so lefties are independent. Anyways, he was looking at handedness. I designed an experiment to investigate the possible interaction between ear stimulated and handedness in an auditory reaction time task. And I think in the reading they talked about that experiment where they said you were wearing headphones and a voice would say left or right, either in your left or your right ear. So that was the original experiment. So he continues, "I was disappointed to find that the predicted ear stimulated by handedness interaction was not significant." So what he went to look for, he didn't find. There was, however, another effect totally unexpected, but as it turned out, much more interesting than my original concern. And that effect, so reaction time was markedly faster when the right command was presented to the right ear than was presented to the left ear. And similarly, reaction time to the left command, so the voice saying left, was faster when it was presented to the left ear than was presented to the right ear. Obviously, an irrelevant cue, the ear in which a participant heard a command, was affecting the time required to process the symbolic content of the command. They realized almost immediately that it stumbled upon a potent behavioral phenomenon that no one had observed before, a tendency in humans to react to the source of the stimulus. So quite the serendipitous finding, which he continued to investigate for a good deal of his career, actually. The one reason the Simon effect is so famous is because it really helped investigate mechanisms of cognitive psychology beyond stimulus response compatibility. We'll talk about it more in that framework, but it has been used in many different tasks to study things like attention. I'm trying to think of other good examples. How we form response codes, like some very nuanced things within cognitive psychology. So there was a conference held to kind of honor the Simon effect and J. Richard Simon back in, I don't know if the conference itself was 2010 or 2011. I wasn't there. I'm trying to think, I guess, was I into research by then? I guess I had gotten into research, but I don't think I even knew about the Simon effect at that time. So they held a conference, I think it was at the University of Iowa, where Professor Simon spent his career, and people kind of from around the world came, and it was a big deal. It was a big event, because a lot of the times discoveries, might not find momentum until after the discoverer has passed away. But in this case, Professor Simon was alive and researching, so they could have a conference in his honor. So that was the name of the conference. And then from it, they published a special article in Acta Psychologica, so a cognitive psychology kind of journal. And all the articles in that issue were about the Simon effect. So it's 10 years ago now, but it's still a great resource summarizing 60 years of research on the Simon effect. There's a lot of good articles in that special issue. One that's fairly accessible is by Professor Hommel. I think, not entirely sure if I'm pronouncing his name correctly. He's a juggernaut in the field of cognitive psychology and has done some work on the Simon effect as well. So as I said before, that the Simon effect is expanded to a lot of cognitive psychology. He mentioned that in his abstract here. So the highlighted part, says the Simon effect can look back at a long and varied history. First treated as a curious observation with implications for human factors research, we'll kind of focus on that today. It slowly evolved not only into a valuable target of psychological theorizing itself, we'll look at that too, but also into handy means to investigate attentional operations, the representation of space of one's body, the cognitive representations of internal action and execution control. But we'll focus mostly on psychological theorizing about how the Simon effect mechanistically occurs within the mind and the brain. Another interesting thing about Simon, this was in one of the articles in that special issue, is we can draw or we can track his lineage back to kind of the founder of experimental psychology. So here's Professor Simon, and he was supervised by Professor Smith. So I don't know if you've heard this before in academia, but we would call this is his academic father. This is his academic grandfather. So let me get my notes here. So I don't remember the details on all of these. So Simon, of course, the originator of the Simon effect. His advisor was Professor Smith. He actually was one of the early researchers in human factors. So kind of like Paul Fitts. Now, Carl Smith, his supervisor was Professor Carmichael. Carmichael, let's see. Oh, he researched primate behavior. So a little bit different. His supervisor was Professor Dearborn, and he's famous in cognitive psychology, but looking at more like child development and its applications to education and how to teach. Now his supervisor, so a father, grandfather, great grandfather, great 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 grandfather. So we're going back a few years here. This is Professor Cattell. Now he is pretty super famous. Sounds like an oxymoron. He was the first professor of psychology in the US. So that's, a big accomplishment. He was the editor of the journal Science for nearly 50 years. And science back then had a huge impact factor, and it still does. So the impact factor of the journal Science, at least last year, was 42. So that means if you publish there on average, you'll have 42 citations to your work in the next 2 or 3 years. I forget exactly the formula. 4 years, next 4 years. So that's huge. So I typically publish in journals that maybe have an impact factor, between 1 and 3, typically. So Science is one of the biggest journals in the world. Now his supervisor was Wilhelm Wundt. I think that's maybe how it's pronounced. German? I don't know now. European, we'll go with. So Professor Wundt is known as the father of experimental psychology. So, psychology as an experimental science, began with him. He actually started the first psychology lab. University of Heidelberg. I wonder if that's German. Does anyone want to Google that? See if it's German. But then he later came to Harvard. Oh, wait a second. No, he got his PhD in Germany, and then he was a professor at Harvard. I think eventually, he was a professor overseas for a long time. So that's quite the academic lineage. And Professor Wundt was one of the first people to do reaction time research. Heidelberg is a city in Germany. Okay, I thought he was German. So would it be pronounced Wundt then?
Probably Wundt. Total guess. We would know. Linvold, you would know. That's my last. Yeah. It's German.
Yeah, and how do you pronounce it? I'll have to go to Germany and work on that. Can you trace your lineage back to Professor Wundt? All right, well, that's enough of our history. It's kind of interesting. 6 generations. In some ways, not too long ago. So this is the Simon task, or at least this is called the standard version of the Simon task. There are a few versions of it. So the one where the first one that was ever done was the speech left or right, into left or right here. And that is a little more, well, we'll look at it, but there's more dimensional overlap in that task. We're going to go to the standard Simon task, which is a little easier for participants. There's less dimensional overlap. We'll get into that soon. So on the monitor, so think of this like as the screen, you're going to get one of 4 stimuli on each trial. And I made this the same as the one that you'll do in the lab. So there are different variants that are still a standard Simon task, but here we'll have color. So is the stimulus red or is it blue? And then we're also manipulating, does the stimulus appear on the left side or the right side? And what you're told is the relevant stimulus here is the color. So the instructions are, if you see a red stimulus, press the left key. So the red stimulus could be on the left or the right, but the position of the stimulus is irrelevant. It doesn't matter to the task. So red stimulus, left key, blue stimulus. So the relevant thing is the color. If it's blue, press the right key. It doesn't matter actually which side it appears on. And from what we know about compatibility, these are probably going to be compatible, easier, shorter reaction time. And these are incompatible, longer reaction time, more errors. And what we're seeing is that your brain can't help but process that irrelevant information of the stimulus side. Even though technically it doesn't matter, who cares if the red is on the left or the right? Your brain says, I do care. And if the red is on the same side as the key press, you're going to be faster than if it's on the opposite side. And you'll get to try it in the lab, and that will likely be the case.
So this is still the Simon that it's not auditory?
Yep. There is an auditory version of this, but they, of the standard Simon task, but they don't say left and right. They'll have a high pitch and a low pitch. And that's to decrease the dimensional overlap, and that's coming up. So we can interpret this with the dual process model. That's the full form of it, but let's just go to an applied situation, so it'll be more meaningful. So let's look at one of the compatible tasks. So we could have either looked at red, press left, or blue, press right. Same sort of processing in both cases. So we do some early stimulus identification, and then the automatic pathway says, well, I saw something on the left. The most natural automatic thing to do is to press left. The intentional pathway says, okay, well, hold on. What were the instructions? It's not about sight, it's about color. So I need to know what color it is. It's red, and the instructions are if it's red, press left. So the intentional process wants to press left, the automatic process wants to press left, and it's already actually programmed that response. They come together. Hey, do we want to do the same thing? Yeah, we both want to press left. You already prepared it. So let's go ahead and initiate that. We have one step here instead of 3, so we have a shorter reaction time. In an incompatible trial, so this is an example of red is on the right, you have to press left. We could have also looked at blue on the left where you have to press right. And here I'm trying to show what the automatic pathway wants to do. So it wants to select. Did I mix up my colors? Oh, I mixed up my labels, didn't I? Oh, excellent. My apologies. So because the stimulus is on the right, the automatic pathway says, I want to make a left response. I'm going to program a right response. And then I guess, I think I have the automatic on the top, but they still want to do opposite things. Okay. So automatic pathway, hey, there's something on the right. I want to press right. I'm going to program right. The intentional pathway, it says, okay, think about the instructions. It's about color, not position. And when it's red, the instructions are to press left. So I'm going to select a left response. Uh-oh, we want to do different things. We're going to trust the intentional pathway. We know it's thought more deeply about the instructions. So we're going to have to abort the response. Oh boy, change that. We're going to have to abort the right response from the automatic pathway. We're now going to have to program the left response, what the intentional pathway wants to do, and then we can go ahead and initiate that response. I'll update these on Blackboard. So that's 2 more steps than we saw before, so we have a longer reaction time. Do we have any questions about applying the dual process model to the Simon task? Okay. Now let's look at the Simon task and a few other compatibility tasks in a little more depth. So the dimensional overlap model is kind of an extension to the dual process model, and it's kind of getting at a lower level of explanation for why there is interference in these tasks. So the dimensional overlap model was spearheaded by Professor Kornblum, and when I was Googling it, I actually found that he has a website dedicated to just dimensional overlap where he explains a lot of the tasks. So it's actually quite helpful. It's more approachable than, say, the literature. If you'd like to check out more information, it's quite well done. So here's the standard Simon task. That's the same slide as we saw before. And we need to think about 3 things. So we'll do this for a few different tasks. But the first question is, well, what's the relevant stimulus dimension? And for this version of the standard Simon task, it's color. You're told to respond based on color. So that's the relevant dimension. In all these tasks, we're also going to have an irrelevant stimulus dimension. And in this case, it's position or horizontal position, left or right. It's irrelevant, doesn't actually matter. Then the third thing we need to look at in terms of the task is, well, what about the response? How is it situated or organized? And in this case, I'm trying to think. In most of the examples we're going to look at, it's a left or right response. So the response dimension is, a left or right key press. So you can describe, we'll look at maybe 4 more examples, but we're going to describe these 3 features of the task. In terms of dimensional overlap, and this is a way to think about how difficult is the task, or how many things are interfering with each other. And in the Simon task, we know that the irrelevant information is interfering with the response dimension. And that's what the dimensional overlap is going to conclude, but with some helpful terminology that helps assist us when looking at other tasks. So the first question is, now that we've described the task, is there overlap between the relevant stimulus dimension and the response dimension? So we're going to compare these 2 things. So relevant stimulus dimension is about color, response dimension is about left or right. Do those overlap? No. And by overlap, I mean, are they the same or similar concepts? And color and left and right, well, those are different things. They're apples and oranges, right? Yes, I don't know if apples and oranges are the best example because they're both fruit, which would have some overlap. But they're, apples and motorcycles. So they don't overlap, so the brain, isn't confused by those 2 concepts. So we'd say no, there's no overlap there. Now, is there overlap between the irrelevant stimulus dimension and the response dimension? Well, yes, that was the one we mentioned right away. It's confusing. The irrelevant stimulus dimension interferes with the response dimension, or in other words, there's overlap between those 2 concepts because they're both about left or right position. So yes, there is overlap here. And when there's overlap, that can potentially make the task harder. Is there overlap between the relevant and irrelevant stimulus? So, well, the relevant stimulus is color, the irrelevant stimulus is left or right. And again, that's like apples and motorcycles, they're different concepts, so they don't interfere with each other. So no, there's no overlap between them. Oops. So any questions about describing the task and then looking for overlap?
So whenever we're doing it, so we're sitting at the keyboard, is it going to be a keyboard?
Yep, two buttons.
2 buttons. So is it going to be to where either a blue light shows up or a red light shows up?
Yep.
So it's based on color.
Yep, yeah, that's what the one I went with this exact example because that's what we'll do in the lab.
So it'll say something like, press the button if it's only red. And then a blue light will show up, but we don't press it.
Oh, no. So the instructions are: if it's red, press the left button. If it's blue, press the right button. So you always make a response. It's whether it should be a left response or a right response. You could do it as a go, no, go. I'm not quite sure how it would work out, but that could potentially work too. So a little description. These different types of tasks fall into what Professor Corwin calls these ensemble types. This happens to be ensemble 3. And the way we describe it is that there's no overlap between relevant stimulus and response. There is overlap between the relevant stimulus and response overlap. In this case, it's the left and right side and the left and right response. There are other ways to achieve that overlap. And is there overlap between the relevant and irrelevant stimulus? No, because it was color and sign. And an example task is the standard Simon, so this version of the Simon. But there are other ways to achieve this with different types of stimuli. When you see that, you might be thinking, well, there's got to be other types of tasks where maybe the answers are yes, no, no, or no, no, yes, or well, what about yes, yes, no? So how many different combinations can we have if there are 3 distinctions and each one is a yes or a no? 6 Victoria? No, 8. 2 to the power of 3. So 2 times 2 times 2, right? So here they are. So there are all sorts of tasks that we can design to create dimensional overlap in 1, 2, or 3 dimensions of the task. So our typical compatibility, this would be our ipsilateral and contralateral mapping. It is a yes, no, no task. And we'll look at that just to reinforce that knowledge within this framework. We'll look at the flanker task. There's versions of this. So again, it's the standard flanker is a no, no, yes task. These middle ones are kind of weird. If you look at how this one's actually not so weird. This one's really weird. And this one's curious. I haven't thought about this much myself, but it's suggested it's impossible. I'll agree with the last 60 years of research. They haven't been able to do that one yet. And then we will look at this last example too, the Stroop task, or the original Simon task, the one saying left or right, actually has overlap in all three of those dimensions. And we'll look at Stroop at the end as an example. And this is something that cognitive psychologists get very excited about, because now you can, for example, compare, well, they've done it a little better than this, but you could do these three tasks, look at the costs involved, and it would give you an idea of how much cost there is to this overlap versus that overlap versus this overlap. And then you maybe predict that those costs might be additive. So let's say this one is 10 milliseconds, 20, and 30. Well, if we do this task, will those costs add together? And will we see a 60 millisecond cost here when we combine all three? So that's the sort of thing that cognitive psychologists, reaction researchers get excited about. You're all thinking, I do not want to be a cognitive psychologist. All right. So we're gonna look at compatibility. So we've done this before, that was our last lab. This is the ipsilateral mapping. So left stimulus, left response, right stimulus, right response. Very easy task. So the relevant stimulus dimensions here, you're told to respond based on the side. Is it left or right? The irrelevant stimulus dimension, do we have an irrelevant stimulus dimension? Oh, there is none. Okay, I was getting confused. I was like, what's irrelevant about this task? The response dimension, again, is a left or a right key press. So is there overlap between the relevant stimulus dimension and the response dimension?
Yes.
Yes. They're both left or right. And in this case, it won't make the task harder. It actually helps the task. But when we do the contralateral mapping, that overlap, then left stimulus, right response, that overlap makes it harder. So overlap is not necessarily bad unless you're told to respond kind of in the opposite way. Is there overlap between the irrelevant stimulus and the response dimension? Well, no, because there is no irrelevant stimulus dimension. And then again, between the two stimuli, well, there's no overlap here because, well, there is no irrelevant stimulus information. But that is how we get a yes, no, no task, or ensemble number 2, I believe it was. We can do the same thing with the contralateral mapping, but we're going to get the same responses. It's still a yes, no, no. But now when you're responding, you're fighting against that overlap. You're trying to do the opposite. So that's why it's slower. But both of those, ipsilateral and contralateral, fall into ensemble type 2. So let's look at flanker. I think we do the flanker. Yeah, this is the flanker task. This was designed by Professor Erickson, and it's actually fairly famous as well. Not more famous than Simon, but it's up there too. So here in this task, we won't try this one in the lab, but what you're told to is respond based on the middle letter. And in this case, if it's S, make a left response, and if the middle letter is H, make a right response. And you can kind of see what's happening here. Again, I put the compatible conditions here and the incompatible conditions down here. So if you get 3 S's, well, those flanking letters, the flankers, they're not going to be a problem, right? Because they're all S's, so it doesn't matter which one you look at. That's you know it's an S, make a left response. But down here, you have to kind of tell your attention, don't get confused by those H's. I need to respond to the middle letter and not the flankers. And this actually makes you slower when there's that competing information in those flanking letters. And it's kind of interesting because you look at this and you say, well, you know, that's very similar to the Simon task. And yes, it is, but the overlap is, you know, different enough that it falls into a unique ensemble type. So relevant information is the middle letter. You need to respond based on the middle letter. What do you have to ignore or what's irrelevant? The flanking letter. It doesn't matter what those are, you shouldn't do anything about it. The response dimension, again, as we'll see in almost all examples except Stroop, is it's a left or right key press. So is there overlap between the relevant stimulus dimension and the response dimension? No. This is a letter and this is a left or right response. Is there overlap between the relevant stimulus dimension and the response dimension? No, because this is a letter, this is a left or right. Those are distinct things. Is there overlap between the irrelevant and relevant stimulus dimensions? Yes, because they're both letters. So that's what they share in common. So a very specific theoretical construct to explain why one condition is harder than the other.
So all these experiments, you're done in like one single condition where you respond to all the different stimuli in one block, or are there different blocks where you have or don't have all the stimuli?
Sure. Well, the standard flanker task, a block would have these 4 types of trials. Yeah, if you ran this as a block and this as a block, it might still work. I think you're going to see more interference when they're all combined. But you would do, you know, this is one experiment. You know, this is a totally different experiment. Those wouldn't be combined. Right. Oh, Stroop. Have we talked about Stroop before? Yeah, we saw in the reading too last week. Okay. So this is a difficult Stroop trial. Sometimes the color of the text and the name of the word are the same, and that's not very hard. But let's look at the difficult situation. So remember, the automatic pathway in the dual process model likes to read. You know, it's maybe your brain is impressed you learn to read. I mean, it's pretty impressive historically speaking. So you know, you see this, you want to say blue. That's the automatic potential pathway system. So slow down there, you know, literate. We want to say the color of the text, which is green. So the correct response would be, if it were a vocal version of the Stroop task, would be saying green aloud. So the relevant stimulus dimension here is the color of the text. That's what you need to respond to. The irrelevant is the word that you're seeing. It's a color word. You're told, you know, don't read that. And the response dimension, well, this case you have to say the color. So is there overlap between the relevant stimulus and the response dimension? Well, they both involve color. So yes, there is overlap. Is there overlap between the relevant stimulus dimension and the response? Well, the irrelevant is color and the dimension is color. So yes, there's overlap. And is there overlap between the relevant and irrelevant stimulus dimensions? Yes, because they both involve color. So one reason the Stroop task is so hard is because there is overlap between all 3 of these dimensions. And that's another great reason it's a good one to show in class, because there's so much interference that when we give people the difficult trials, they can tell, oh, you know, I'm super slow at this. I typically in my undergrad class have my students try stimulus response tonality ipsilateral contralateral, and they usually feel a bit slower on contralateral. It is 100 millisecond effect, which is quite big. But we also try this, and that one, you know, you can tell for sure that you're way slower when the text, color, and the word are incongruent. I think I added in a standard choice reaction time task. And so when I think of choice reaction time, I usually think of ipsilateral, but there is an even more standard way to do the task that involves no dimensional overlap. Yes, okay, I included it here. This is one example. I kind of like it because it's kind of like the flanker task, but without the flankers. And we've added in an irrelevant dimension, and here it's color. So here your instructions would be if you see the letter S, press the left key, and the stimuli are always in the middle, so there's no left or right stimulus shenanigans in this example. And if you see the letter H, it doesn't matter if it's red or blue, just if it's H, press right. So relevant stimulus dimension is, well, what letter do you see? Irrelevant is color, doesn't matter what color the letter is. You can think about this being irrelevant by, don't even have to mention that in the instructions. You just say, okay, if it's S, press left, if it's H, press right. And as they do the task, it might be, oh, they're different colors, but it's irrelevant. It doesn't matter to the instructions or to how you're supposed to respond. And the response dimension, back to our typical, it's a left or right key press. And when we look for overlap here, we're going to see that there is no overlap. So letters versus left or right, those are different concepts. Color versus left or right, those are different concepts. Letter versus color, again, different concepts. So this sort of task is often used as a control. This is where you should have the shortest reaction time because there's no overlap and also there's no competition between overlap like there would be in the contralateral map. So that's our tour. We didn't look at all of them. That one's fairly straightforward. I don't know an example of this one. This one is super weird, but I've probably shown you too many already. So any questions about those ensembles? So you don't need to, in your presentation, go into that sort of depth on the ensembles. It's okay to just talk about, the standard Simon test. That's the one we're going to try. But I wanted to give you the theoretical background and how there can be overlap in different cases. So let's talk about your assignment. There's going to be some nuances this week, some changes, and I'll explain why. So we're not going to change our deadlines, our typical procedure. So data collection, as I think always, will be due 2 classes later, Wednesday, Monday. Yeah, so you have class Wednesday where you can do data collection, or Tuesday is fine, or Thursday or Friday. And then we'll have another data collection class on Monday, but data collection will be due by the end of the day on Monday. And then let's see, then we've got a Wednesday class for preparing presentations. And then the following class will be the day for student presentations. So that's our normal, 5-class cycle. Data collection takes 15 minutes. There's 2 blocks. We'll talk a little bit about that in a moment. Each block takes 6 minutes. And the Stroop, the Simon task, is a bit cognitively demanding. So I usually give myself, a couple minutes between blocks just to, rest. I guess it's not physically demanding, but it's kind of mentally demanding. But you can, if you're scheduling a time slot, 15 minutes should be enough to get in, do your data, and finish. All right, so this module, so last module, I asked you all to present individual data, which was great. And the one reason I asked that is because the stimulus response compatibility effect is large. It's 100 milliseconds. You typically see it in everyone. And I think we saw pretty good compatibility data in everyone. The Simon effect, in contrast, is not as large. So I've used it in undergrad classes before. And if you look at any individual's data, it's not necessarily, a nice X. It's not going to be a big X, but it's also not necessarily a little X, because there's sometimes variability. Sometimes people get this or that, kind of all over the place. So this module, instead of presenting your individual data, I want you just to present the class mean data. In the past, I've sent you the class mean data to, sometimes you had to show it, last module was optional, but this time, that's what I want you to present, and you're actually going to be calculating the class mean data yourself. Now you might be thinking, okay, do I have to do what I did last week 12 times? The answer is no, you don't have to. You can. That's one approach. You could analyze, all 12 data files. The other thing that we'll kind of use the rest of the class for is you can use a fairly powerful feature in Excel called, do you know where I'm going? Pivot tables. Has anyone used pivot tables before? Have you used them before?
Sort of.
Sort of. So the good news about pivot tables is they're fairly user-friendly. Also, we don't have to go nuts with them. We just need kind of basic data out of them. And I'll show you today pivot tables in action. And basically, they allow you to get data from all the participants, maybe even faster than how we used to get data from just one participant. So come presentation time, I want everyone to present the mean class data. And two things, I want you to present mean reaction time. And I'll show you an example. I tested myself 4 times just to get a little more data than one participant. It's probably not best to do one person 4 times. 4 people once is probably better. But I'll show you my data. And the way we plot Simon task data is actually with the same plot you did last week, the typical stimulus response compatibility plot with reaction time on your y-axis, response location on the x-axis, and then in the legend stimulus position. So you'll plot, there'll be 4 data points. There'll be 2 types of compatible trials. So there's that compatible trial. So you'll get a class mean for this, a class mean for that, and you'll also get class mean for the incompatible trials. So let's see, this one would be a right stimulus, left response. That's that specific one. Last week in the presentations, a common thing I saw, so if you remember the plots, they should look like this. Several people, I think maybe 4, these 2 data points, they mix them up. So be very careful when you're getting your data to think about kind of where they should go in that 2x2 table. And it is confusing. It's something even as I was plotting, I had to go really slow and think about, okay, which data point is this? Where does it go on the table? The other thing, like last module, is I'd like you to present the mean number of recycled trials. So exclude practice trials and then look how many recycled trials you have. We'll just take all recycled trials. We won't worry about whether it's you responded too fast, you didn't respond, you responded too slow, or you pressed the wrong side. We'll just group those all together. And I think this might work out a little better than last time, although with Chase's data, that it did help us last time get a difference between the conditions. So I'm not entirely sure what we'll see, but maybe it will show a nice difference. And the prediction would be that you would have more errors on the incompatible trials than the compatible trials. No, that's right. Okay. So how do we do pivot tables in Excel? Well, first the data file so I won't be able to send you the data file until everyone's done the experiment. So likely on Tuesday, October 26th, I think I remember the date correctly, I'll be able to send you, there'll just be one long data file with every single participant. So I've added a few things to what we've seen before. So the first column will now be the participant number because we need to know who's participant 1 and who's participant 12. So that will be in there. Block number, the way the experiment works is there's 2 blocks and they're actually the same, but you just get a little break in the middle. Each block is 80 trials. So it starts with 16 practice trials that you warmed up, and right into 64 test trials. And then you're going to do that again a second time in block 2. So there's no counterbalance. Everyone's going to do block 1 and then block 2 because it's just the same thing, just to give you a little break in the middle. Because 160 trials on the Simon task, it helps have a little break in the middle. So you'll have the block number in there, then you're gonna have the trial number. It will potentially go to 80. If there's recycled trials, it might go a little higher. The 4-period duration, which is 1 to 2 seconds. Then this tells us what type of trial it is, but we need 2 independent variables to set the stimulus. So which side is it on? 1 is for left, 2 is for right. And then which color is it? Is it red or blue? Let's see, I have a red pen here. So I think I can give an example. So let's see. Let's say the red stimulus appears on this side. This is one of the 4 types of trials. So this type of trial stimulus side, well, it's on the right. So it's going to be a 2. And the stimulus color is red. So it's going to be coded as a 1. So this trial is a 2-1 is another way to think about it in terms of Excel. Did I get those numbers right? And if the red was on this side, then it would be a 1-1. So you'll see as you pull these out, your compatible trials are 1-1 and 2-2. Those are the easy conditions, and the incompatible ones are 1-2 and 2-1. So we've got 4 types of trials. Then we've got, is it a practice trial? So the first 16 of each block, that will be set to true or 1. And then like before, reaction time for the left hand and right hand. If you press that button, you'll get a value, the reaction time in milliseconds. And if you don't, it will be NaN for not a number. So on this trial, hopefully they make a right response. So hopefully when we look at the reaction time, it's something like NaN for the left, because you're not supposed to press left here, and let's say 300 for the right. But if you press the wrong button, if it's 400 and NaN, then the program would recognize you pressed the wrong button, and it would then recycle this trial if it's a test trial, and it would set that to true. Something else I added, because as I was doing it, sometimes you don't realize when you make a mistake. And I think we've had that with a few other people when they were doing the task. Notably when I had the stimulus box upside down. Was that with you, Shelby?
That was Kelsey.
It was Kelsey.
I was in the room, but that was Kelsey.
That's why she's not here. It's dramatic. So what happens if it's a recycle trap, the computer will actually beep at the end of the trap. It kind of gives you feedback of, no, that was wrong. It's not a super loud beep, but you'll probably hear it. The questions about the giant data file that I'll send on Tuesday at the latest. So this is an example. So I did the task as participant 13, 14, 15, and 16. I won't include this in the data file, but you can see here, what you'll expect. And there's a lot of cells here. So there's all we can see is participant 13, the first block, and only my first 30 trials. But that would go, to 80 plus recycle, then block 2, then participant 14, block 1, and so on. So let's see, pivot tables. There's a few slides here about it if you need to refer back to it later. But what we're going to do is I'll walk you through this in Excel. So let's see. Okay. Time and recovery. No, don't need to do that. Sure, just in case. All right. So here are the data. And the first step is to make this an official Excel table. It makes it a little easier later on. So, make sure you're clicked somewhere in the data. Probably the top cell here is the best idea. And you want to say insert and then table. So this is just going to turn this into an official table in Excel. Right now, it's really just a worksheet. So say, hey, make this a table. And Excel is smart enough to say, oh, I think this is your table. And we just say, yes, it thinks there are headers, which there are. So we just say, okay. And there now it's an official table. And the easiest way to tell is because it throws on a style there, but you can change this if that format is not to your liking, but it is just cosmetic. So we now have a table, and just to show you in the slides here. So, oops. So we had this sort of data, and then we said, I want a table. The other thing we're going to do, we're not going to make a pivot table, but that's kind of all the searching you really need to do in Excel. Okay, so we want to make sure we're clicked somewhere on the table here, and now we're going to say insert pivot table. So turn our table into a pivot table. We click on it, and it says, okay, well, it's pretty smart. So it's the table it made it by default named it table 1, and it says I'm going to make a table out of table 1. I'm going to put it in a new worksheet. So we're going to have a new tab at the bottom. So those are all the default options. That's great. We'll say okay, and boom, there we go.
So it is ready to make a custom pivot table with our data. So what we want to build is, just draw it kind of conceptually over here. Because it takes a little dragging and clicking, but if you think about it conceptually, it might make it a little easier. What we want to end up with is a table where each row is a participant number. So in this case, we've got 13, 14, 15, and 16. With the other data, it would be 1 to 12. And then what we want for the rows are the different conditions. So these would be the averages across all those conditions. So we want to know for a 1-1 trial, what is the average reaction time? Or I should say the mean reaction time. And then for a 1-2 trial, a 2-1, and a 2-2. We want to know what these mean reaction times are. So not for every individual trial, but for participant 13 across both their blocks, all their trials, what is their mean reaction time? So we want to tell it to make this sort of table. Now there's two types of trials we want to exclude. We want to exclude practice trials. So this is when that practice trial variable or column is equal to 1. True. Yeah. And then we also want to exclude recycled trials. Now later on, you'll have to make a different sort of table when you want to count the number of recycled trials. But for now, we want to exclude them. So recycled trials, we want to keep ones that are 0, so not recycled trials. And that is kind of all we need to do to make our pivot table. So see down here, it's saying these are the columns in your table. It's automatically identified those. And it says, okay, well, how do you want to make a pivot table, which is like the summary of that table? Well, rows, we want that to be participants, right? Each row is a participant. So I'm going to just drag participant number down into here. There we go. And it knows that, it's smart, right? It knows that there's only participants 13, 14, 15, and 16. With the data table I send you, it'll know that there's participants 1 to 12. All right, well, what do you want in the columns? Well, we want two things. We want to know the stimulus side and the stimulus color. So we drag those in there. And now we start to have these placeholders for where we're going to put in our data. Reaction time, we want to know, depending on the trial, we want to know either the mean of the left trials or the right trials. So that's the values. That's, what's going to go into the table. What's going to go here is whatever we put here. And you can see it's defaulted to count. So we'll change that later to the mean. But right now it's counting how many trials there are of that type. So participant 13, how many trials are there with stimulus side of 1 and a stimulus color of 1? So that would be a left red trial. Well, there are 44. This participant has 44, this one has 42, this one has 40. The reason they're different is because of the number of recycled trials. So let's add in a filter here. I like to leave this as a count just to help me understand whether or not it's grabbing my data properly. So we remember we want to exclude practice trials. So we're going to put that in as a filter, and we're going to put in recycled trials as a filter. And then to edit those, I like to go over here. I'm not sure if you can do it here, but it says, okay, practice trials. Well, which ones do you want? Do you want all? So currently it doesn't matter what that variable is, it's including everything. Or do we want only practice trial 0 or only when it's 1? Well, this would be practice trials when it's 1. So let's do that first and see. Now it's telling you that, oh, well, everyone has 8 practice trials in each of these conditions. And that's exactly what we should have. But we actually want to exclude practice trials, so we'll set that to 0. So the trials have dropped a bit because we're now excluding practice trials. And the last thing we want to do is we want to get rid of recycled trials. So we want to know when they're not recycled. No, recycled practice trial false, recycled trial true, false, right? Why does it seem weird to me? Did I write the wrong thing on the board? I wrote the wrong thing on the board. Practice trials. Do not want practice trials. Do not want practice trials. We do not want recycling trials. So now you can see everyone has the same number of test trials, 32. There's 32 trials in this condition, and then this is a left stimulus, blue, 32 trials of those. In gray here, this is actually collapsing across both these factors, so it adds them together. We're actually going to kind of ignore those fields. But then over here, you can see that, well, we've got 32 trials in that condition and 32 in there. So that's okay for counting. The counts add up. But what we want to do is we want to see the mean reaction time. So we click on this and we just change the value field setting. Right now it defaults to count, but we want an average, which is a mean, just that's the loose terminology that Excel uses. And we'll change this one to setting average, which is a mean. There we go. So here we go. For a participant 13, stimulus on the left, in color red, the mean reaction time across those, 32 trials is about 410 milliseconds. Now, why is there no value here? Well, that's because you never actually press the right button. That would be the wrong response. So every trial gives you a left and a right response, even though you're not pressing that button. So what it tries to do here is it's trying to add 32 trials of NAMs divided by 32, and it dies. So we don't have a value here, and that's okay. We do have a value here, so this is a left stimulus blue, and remember when it's blue, you should press right, so we do get values here. So this is a compatible trial, that's the incompatible trial. And the data already look pretty good because the compatible is faster or shorter than the incompatible. And now our other two data points are over here. So this is a 2-2 trial here. So these are the individual participant data. Now to get this value, this is the mean of those means, or what we also call a grand mean. So these are the data points that you want to plot. So that one, and then these two over here. So those are your 4 data points that will go into that 2x2 compatibility table. So what are your thoughts on this? Does it seem crazy? Just seem weird?
A lot of steps.
All of this to avoid MATLAB.
Yeah, this is really the intermediate between Excel and MATLAB. So I mean, you can imagine, you know, if your boss in your future career comes to you and says, "Hey, we've got 100 tables, and you know, you don't want to analyze everyone individually. You want to throw them all together and then, you know, pivot table that bitch." That's what you want to do. Now, what I would suggest, and this is something I did, is because when these values come out, it's like, oh, are they correct? I don't know. Well, one thing that helps is leaving it as count, so you understand that, yes, it's got the right number of trials. The other thing I did do is for one participant, I think participant 13, I did it the old-fashioned way, where I went through and calculated the data. Let's see, do I still have it here? Let's see if I have it here. Nope. I don't remember exactly which. There we go. So for 13, I went through, removed the practice trials, removed the recycle trials, sorted the data so that these are all trial 1-1s here. Down one further. And then I calculated the reaction time and see, so it's 409.875. And then I could compare that to see if, you know, my magic pivot table actually worked. And yes, it did, 409.875. So you might want to do your own data individually the old-fashioned way, and then do the pivot table and make sure your data is replicated. And then if yours is right, then hopefully everyone else's data got pulled into that pivot table correctly. So eventually I made a little 2x2 table. Let's let's see here. This one. So I made a little 2x2 table with the left response, right response, left stimulus, right stimulus. And I went and I pulled out those 4 points and just copied them and pasted them into here. And then we came up with a nice stimulus compatibility graph for the Simon effect. And it's not perfect. It's not a super nice X. The Simon effect, there's a picture of this in the textbook, and I think the magnitude or the difference between compatible and incompatible is usually like 30 milliseconds when you average across participants. So here I'm seeing a pretty big size. This is the compatible trial, so that's good. That's my incompatible trial, so that makes sense that this is slower than that one. It's a little bigger than normal, which is okay. But then on the other side, so this is compatible, that's incompatible, so that's the right direction, but that's only 10 milliseconds, and it should probably be 30. And the Simon effect isn't great on any one individually, so I wasn't too surprised to see this. But hopefully, once we pool 12 participants together, we'll see something similar to what's shown in the textbook. Also, my number of recycled trials, you don't need to separate into 4 groups. You can just see how many are compatible, how many are incompatible. And I mean, there's a slightly more in the incompatible block, but didn't work out too well for me. Hopefully with 12 participants, we see the predicted direction that there's more mistakes on compatible trials than incompatible trials. So just a reminder of our due dates. Any questions? We have a little time left, so I thought maybe we'll just we'll go to the lab right now, and we can just fire up the computer and you can see it all in action. And then you'll be ready to collect whenever you want. I'll leave my stuff here, but if you'd like to come to the lab, we can convene there in just a couple minutes, and I'll show you the code.
I got this from the Walmart.
Oh,